Hello everyone, I'm Eber. Today we will be examining if 8 is really the optimal pH for trypsin. So you might ask yourself, what is an enzyme? An enzyme is a catalyst that lowers the activation energy needed for reactions to occur. It does so by forming the bonds of substrate molecules and creating microenvironments that promote the reactions. Most enzymes are globular proteins, and you begin the catalyzation of reactions when an enzyme binds to a specific substrate molecule at an active site. This binding creates conformational change in the substrate molecule that in turn lowers activation energy. This presence of enzymes is so critical to human life because without them, the chemical reactions in our body would not proceed in a fast enough time to meet the needs of human life. This is largely due to the high activation energy chemical reactions in our body require. The optimal pH for the enzyme trypsin to work in the small intestine is 8. The two things that led me to my hypothesis was that different pH levels in the environment could change the conformation of enzymes by affecting the number of their bonds and their locations. If this is affected, they wouldn't be able to bind to substrates, therefore unable to catalyze reactions. This led me to hypothesize that as the pH deviates from the optimal pH at 8, there will be a decrease of enzyme activity during the breakdown of azocation. So the purpose of my protocol was to test buffers at different pH levels and to examine how enzyme activity changes as it deviates from the optimal pH of 8. I first began by gathering 5 1.5 ml clean centrifuge tubes. I filled the 5 centrifuge tubes with 450 microliters of 0.15 molar sodium chloride and 20 tris ECO to achieve a buffer at pH 6. I added 500 microliters of azocasin and 50 microliters of the enzyme trypsin into each of the centrifuge tubes. The 5 tubes were incubated at 50 degrees Celsius for 10 minutes. After incubation, 500 microliters of TCA 10% was inserted into each of the tubes and mixed. The tubes were centrifuged for 2 minutes at 14,000 grams. The liquid portion of the centrifuge tubes was inserted into 4 separate cuvettes. Once the cuvettes were prepared, the spectrophotometer was cleared using deionized water and set at 440 nanometers. Each of the cuvettes were inserted into the spectrophotometer individually, and the absorbance was recorded for each of them at 440 nanometers. After this whole procedure was done, it was done 3 more times for buffers at pH 7, pH 8, and pH 9. The only thing that changed in the procedure that was done again was the buffers. The azocasin was kept the same at 500 microliters, and the enzyme trypsin was kept the same at 50 microliters. It is important to have a control variable without the enzyme trypsin. Trypsin is an enzyme that breaks down the dietary protein. In the case of this lab, the dietary protein will be azocasin. There are cases in which a protein will begin to break down without the assistance of an enzyme. It is critical to have a sample without trypsin because it shows the scientist if azocasin undergoes a breakdown without the presence of trypsin. It will allow the data to be more accurate because it will inform the scientist if the enzyme is the only factor in the breakdown of azocasin or not. The control samples were created and went through the same process as stated in the procedure in the last slide. The only difference is that the 50 microliters of trypsin is replaced with 50 microliters of deionized water for all the. So what we have here are the averages for our control samples. And as you can see, at pH 6, 7, 8, and 9, they all have an activity. So what this means is that azocaine undergoes natural breakdown and that azocasin is not only broken down by trypsin. As you can see at pH 6, the average is 1.92. The average for pH 7 is 2.04, and the average for pH 8 is 2.07, and finally the average for pH 9 is 2.04. You might ask, what am I going to do with the control samples? Well, I'm going to use the control samples to find the true values of my trypsin samples. So first, I can take any control sample, but just to show an example, I'm going to use one sample at pH 6, which is my original trypsin sample. I'm going to subtract that against the average for the control samples at pH 6, and that is going to give me the true trypsin value for a sample at pH 6. I can do this for any sample at any pH to get the true value. These results are the results of the trypsin containing samples. This graph represents a relationship of Ng of product per milligram of enzyme per minute and pH 6, 7, 8, and 9. You can see that as the pH increases from 6 to 9, the product produced also increases. If we look closely, we can see that the standard deviations of 6 and 7 do not overlap, which proves that the data sets of pH 6 and 7 are statistically different. If we look at the data set of pH 7 and pH 8, the standard deviations do not overlap, which proves that these two data sets are statistically different. If we look at the data sets of pH 8 and pH 9, the standard deviations do not overlap, which proves that these data sets are also statistically different. And if you remember from my hypothesis, I hypothesized that the highest enzyme activity in the breakdown of azocasein would be at 8. But when I look at this, knowing that all these data sets are not statistically similar, they're all statistically different, this shows me that in this case, pH 9 is the optimal pH for trypsin and in the breakdown of azocasein. For the data shown in the last slide, I conducted a t-test. And with this t-test, it conducts an analysis of two data sets. So as we saw in the last slide, I compared the standard deviations and came out to conclude that they are all significantly different. But what the t-test does is that it takes it a step further and analyzes two data sets. And I know that if the p-value is less than 0.05, then the alternate hypothesis is proven, which is the opposite of what is stated in the first column. So I compared the observance of pH 8 and pH 9, and that came out to prove that it was significantly different. I did the same for pH 8 versus pH 7 and pH 8 and pH 6, which all proved to be significantly different. And what this proves is that the optimal pH for the enzyme trypsin in the breakdown of azocasein is 9 and not 8. So my hypothesis is disproven because if you see in the last slide, the pH at 9 was the highest bar, which proves that that is the highest point in which enzyme activity is present. I would say that my experiment was pretty effective in providing data results. It provided me with the data necessary to prove at what pH the optimal pH was present at. In this experiment, my hypothesis was not proven because it proved that the optimal pH is located at pH 9, not pH 8. The visual representation presented on slide 8 showed that the pH at 9 was the highest with enzyme activity. 9 of the standard deviations intersected, which proved that all the data is significantly different. I went a step further and conducted a t-test, which proved that all the data analytically was significantly different. This further proved that in this experiment, the pH at 9 is the optimal pH for trypsin. It is very important to have a pH that is optimal for all the enzymes in your body, because if the pH is too high and passes the optimal value, it can denurture the enzyme. If the pH is too low from the optimal value, the conditions won't be favorable for it to catalyze the reactions. If I were to change something for the next experiment, I would change two things. I would change the variability of my procedure. I would add pH 10 to the sample pool. I have two samples below pH 8, but only one above pH 8. Adding one more sample would actually prove to me if pH 9 is truly the optimal pH or if it is something greater than that. I would also bring down the temperature of incubation from 50 Celsius to 37 Celsius to reflect the human body temperature. Thank you for taking the time to view my PowerPoint. Before we end, I wanted to give a special thanks to Dr. Margolis for his guidance throughout this experiment. I want to thank the biology department at Towson University for providing the enzyme lamp protocol, and I want to thank James Stegman for providing the statistics intro PowerPoint that helped me with my t-test.